Hello friends, today we will learn about cell transformation and cell cloning. So let's begin. Cell transformation due to change in the genetic material and cell cloning involving the production of the population single cell. Now we will discuss. Now first point comes in our mind is transformation of cells. Transformation broadly refers to the change in the phenotype of the cell due to a new genetic material. As regards the cultured cells, transformation involves spontaneous or induced permanent phenotypic alterations as a result of heritable changes in DNA and consequently gene expression. Transformation of cells may occur due to any one of the causes. Let's discuss about this causes. First is spontaneous genetic material may be spontaneous changed infection. Second is infection with transforming viruses. Third is from gene transfection. Fourth is exposure to chemical carcinogens. Fifth is exposure to ionizing radiations. Now let's know about characteristics of transformed cells. The general let's know about the general characteristics. They are grouped as genetic, structural, growth and neoplastic, and transformation is associated with genetic instability, immortalization, aberrant growth, control and malignancy. Now, bit by bit, we will discuss in this video. First is genetic instability. What is this? In general, the cell lines in culture are prone to genetic instability. A majority of normal finite cell lines are usually genetically stable, while cell lines from other species, example mouse, are genetically unstable and can get easily transformed. The continuous cell lines derived from tumors of all species are unstable. The normally occurring genetic variation in the cultured cells are due to some causes. Let's know about these causes. First is high rate of spontaneous mutations in the in vitro conditions, possibly due to high rate of cell proliferation. Second point is the continued presence of mutant cells in the culture as they are not normally eliminated. Now let's know about immortalization. The acquisition of an infinite lifespan by a cell is referred to as immortalization. Most of the normal cells from different species have a finite lifespan of 20 to 100 generations. But some cells from mouse, most of the tumor cells have infinite lifespan as they go on producing continuous cell lines. Now let's know about control of finite lifespan of cells. The finite lifespan of cultured cells is regulated by about 10 senescence genes. These dominant acting genes synthesize products which inhibit the cell cycle progression. It is strongly believed that immortalization occurs due to inactivation of some of the cell cycle regulatory genes like RBP53 genes. Now let's know about immortalization of cells by viral genes. Several viral genes can be used to immortalize cells. Let's know about these genes. First is SB40LT, HPV16E6 by E7, HTRT, ADR5E1A, EVB. Among these viral genes, SV4OLT is most commonly used to induce immortalization. The product of this gene, T antigen binds to senescence genes such as RB and P53. This Binding restricts surveillance activity of senescence genes. The result is an increased genomic instability and activity leading to further mutations favoring immortalization. For the process of immortalization, the cells are infected with retroviruses containing immortalization genes before they enter senescence. By this way, the lifespan of the cells can be extended by 20 to 30 population doublings. Thereafter, the cell ceases to proliferate and enter a crisis phase that may last for several months. At the end of the crisis phase, a small portion of cells can grow and eventually become immortalized. Now let's know about immortalization of human fibroblast. The human fibroblasts are most successfully immortalized by the viral gene, namely SB4OLT. The process of fibroblast immortalization is complex and indirect with a very low probability, that is about 1 to 1 in 10 to the power 7 cells. Now let's know about immortalization of cells by telomerase induction. The most important cause of infinite lifespan of cells, that is senescence, is due to the telomeric shortening followed by cell death, or this is apoptosis. If the cells are transfected with telomerase gene STRT, the lifespan of the cells can be extended and a small proportion of these cells become immortal. Now let's know about aberrant growth control. The transformed cells and the cells from tumors grown in culture show many aberrations with respect to growth and its control. Now let's know about encourage independence. There occur several changes on the cell surfaces of transformed cells. These include alterations in the cell surface, glycoproteins and integrins, and loss of fibronectin. Some of the transformed cells may totally lack cell adhesion molecules, that is CAMs. The modifications on the surface of transformed cells leads to a decrease in cell and cell substrate adhesion. The net result is that there is a reduced requirement for attachment and spreading of the cells to proliferate. This phenomenon is referred to as encouraged independence. Encouraged independent cells grow in a disorganized fashion. These cells may be comparable with the tumor cells detached from the native tissue, which can grow in foreign tissue, that is formation of metastases. Now let's know about contact inhibition. The transformed cells are characterized by loss of contact inhibition. This can be observed by the morphological changes in the disoriented and disorganized monolayer cells. This results in a reduced density limitation of growth, consequently leading to higher situation density compared to normal cells. Now let's know about low serum requirement. In general, transformed cells or tumor cells have lower serum dependence than the normal cells. This is mostly due to the secretion of autocrine growth factors by the transformed cells. Some Let's know about some growth factors produced by tumor cells. First is colony stimulating factor, in short CSF, transforming growth factor, that is TGF-A, interleukins 1 to 3, vasoactive intestinal peptide, that is in short VIP, gastrin releasing peptide. It may be noted that many normal cells, that is fibroblast endothelial cells, also produce autocrine factors during active stage of cell proliferating. Hence, these factors will not be of much use to serve as markers of cell transformation. Now let's know about tumorigenicity. Cell transformation is a complex process that often results in the transformation of neoplastic cells The cell lines obtained from malignant tumors are already transformed. Such cells may undergo further transformation in the in vitro cultures due to increased growth rate, immortalization, and reduced anchorage dependence. For the malignant transformation cells, several steps may be required. First step is the cells can be cultured from malignant tumors and characterized. Second is viral genes or chemical carcinogens can be used to transform the untransformed cells. Now let's know about cell cloning. In the traditional culture techniques, the cells are heterogeneous in nature. Isolation of pure cell strains is often required for various purposes. Cell cloning broadly involves the processes connected with the production of a population of cells derived from a single cell. Cloning of continuous cell lines is much easier compared to that of the primary cultures and finite cell lines. There are certain limitations for cloning of culture cells derived from normal tissues. These cells survive for a limited number of generations and therefore cloning may not result in any significant number of cells. On the other hand, cloning of continuous cell lines due to their transformed status is much easier. Thus, the transformed cells have higher cloning efficiency compared to normal cells. Cloning may be carried out by two approaches, monolayer and suspension cultures. First is monolayer culture. In monolayer culture, what happens? Petri dishes, multi-well plates or flasks can be used for cloning by monolayer culture. It is relatively easy to remove the individual colonies of cells from the surfaces where they are attached. Second is suspension culture. Cloning can be carried out in suspension by seeding cells into viscous solution, that may be methocell, or gels, that means agar agar. As the daughter cells are formed in suspension, they remain intact and then form colonies in suspension. Now let's know about dilution cloning. Dilution cloning is the most commonly used technique for cloning of monolayer cells. Now let's know about dilution cloning. Dilution cloning is the most commonly used technique for cloning of monolayer cells, and it involves various stages. Let's know about these stages. First is trypsinization. Trypsinization of cells at log phase to produce single cell suspensions. Second, dilution of the cells to about 10 to 100 cells per ml. Third is seed the cells in multi-well dishes, petri dishes or plastic bottles. Fourth point is incubate under appropriate conditions for 1 to 3 weeks. Fifth is isolate individual colonies. The clones can be isolated from the multi-well dishes by trypsinization, by cloning ring technique from petri dishes, and by irradiating the plastic bottle. Now let's know about stimulation of plating efficiency. Plating efficiency represents the percentage of cells seeded at subculture that gives rise to colonies. The plating efficiency and cloning efficiency are said to be identical if each colony is derived from a single cell. The plating efficiency is around 10% for continuous cell lines, while for primary cultures, the finite cell lines, it is quite low, 0.5 to 5% or sometimes even 0. Several attempts are made to improve the plating efficiency in the culture laboratories. These approaches are based on the assumption and that the cells at low densities require more nutrients or growth factors. The stimulation of plating efficiency with regard to culture factors, conditioned medium and feeder layer, now we will discuss. we already discussed. Okay, now we'll discuss about culture factors. First factor is medium. A medium rich is in various nutrients is better suited for improving the plating efficiency. Second is serum. Fetal. Fetal bovine serum is better than horse or calf serum if the addition of serum is required. Now, addition of metabolites. Supplementing the medium with intermediary metabolites, example pyruvate or alpha ketoglutarate and nucleoside stimulates plating efficiency. Now, let's know about addition of hormones. Dexa, dexamethasone, a synthetic analog of hydrocortisone, improves plating efficiency. Example, fibroblast, melanoma cells, chick myoblast. Insulin also stimulates plating efficiency of several cell types. Now let's know about carbon dioxide. Carbon dioxide significantly influences plating efficiency. Most of the cells require 5% carbon dioxide, while for some cells a lower carbon dioxide concentration is better. Example, human fibroblast. HIPS is used to protect the medium from pH fluctuations. Now let's know about pre-treatment of substrate. The plates pre-treated with fibronectin or polylysine show an improvement or improved plating efficiency. Now let's know about use of purified trypsin. Some workers prefer to use purified trypsin instead of crude trypsin for trypsinization so that the plating efficiency is better. Now let's know about conditioned medium. The medium that has already been used for the growth of other cells contain certain metabolites, growth factor and other products that stimulate growth. Such a medium, referred to as conditioned medium, when added to the cell cloning medium, improves plating efficiency. Now let's know about feeder layers. The layer growth arrested living cells, referred to as feeder layer, promotes plating efficiency. This is because the feeder cells provide nutrients, growth factors and matrix constituents that support survival, growth and proliferation of cells. Now let's know about suspension cloning. Certain cells, particularly the transparent fibroblast and hematopoietic stem cells, can be more conveniently cloned in suspension rather than monolayers. Suspension cloning can be carried out by using agar or methocell, which can hold the cells of a given colony together and prevent mixing of colonies. The technique of cloning in agar suspension is carried out in some stages. Let's know about the stages. Cells from suspension culture or monolayers can be used. The monolayer cells have to trypsinized, while the suspension cells can be directly used. Count the cells and dilute serially so that 10 to 200 cells per ml are finally present. Third point is freshly prepared agar medium with appropriate dilution is used. Fourth point is the agar medium is inoculated with the diluted cells. Fifth point is incubation for 1 to 3 weeks given clones in culture. Now let's know about isolation of clones. After the cloning is complete, the selection and isolation of specific cell strains is the next important step. This is essentially required for the propagation of cells. If the monolayer cells are cloned directly in the multi-well plates, the colonies can be isolated by trypsinization of the individual wells. However, when the cloning is carried out in petri dishes, the colonies can be separated from the medium by the placing stainless steel or ceramic rings around the colonies. Now let's know about micro manipulation. The genes of a colony, that is the colony is derived from a single cell, can be done by micro manipulation. This can be achieved by monitoring the colony formation at the early stages of cell cloning. Macro manipulation, if properly done, is believed to be conclusive method for determining the genuine clonality of a clone. Finally finished. Any questions regarding this, you can ask me in the comment box, and subscribe my channel to know more updates. Thank you.